I'm Rachel Simmonds. I am Professor of Immunopathogenesis here at the University of Surrey, where I'm also head of the discipline of microbes, infection and immunity. Yeah, so my lab researches a disease called Buruli ulcer, which is a rare and neglected tropical disease caused by Mycobacterium ulcerans. And our lab mostly focuses on the mechanism of its virulence factor, mycolactone, and understanding how that causes the disease and interaction with the host. So in this particular project, one of my PhD students is looking to measure the growth of the bacteria within macrophages, and there are some challenges around that because the bacteria is high containment, which means that we can't use all the normal equipment we have around about in the lab, and so we've developed a methodology to measure that growth using an operon which we've cloned into the bacteria that expresses a luminescent protein, and so we can measure the growth by measuring the luminescence over time.
My name is Kwabna Boateng. I'm a PhD student at University of Surrey in Professor Rachel Simmonds' group. We are starting to realize the importance of macrophages in the pathogenesis of the disease. So one of the questions that we want to answer is, can the bacteria actually grow inside macrophages? So what we use the MyGlo is we've been able to engineer bacteria to be luminescent. So what we do is we infect the macrophages with these luminescent bacteria. Then we can use the MyGlo to track basically the increase in luminescence over time, which we are using as a correlative marker for growth.
The alternative methodologies are challenging, so in terms of counting bacteria under microscopes, and that we were limited to that because we have to work in high containment. So we essentially using assays that involve things that we can work inside the cabinet or things that involve dead cells. So the MyGlo has basically made it possible to do assays which have been around for quite a long time, where you can measure the growth of bacteria using luminescence. But those assays only work on live bacteria. And so doing them at high containment is a real challenge. So the MyGlo, because it's a small instrument that we can put inside the cabinet, it means that we can do those assays with live cells that we wouldn't have been able to do before. And it's very small, it's not much bigger than a 96-well plate, and has a single wire connected to it, and I could see immediately that it would work very well inside a cabinet in a high containment environment. The major step forward is showing that we can do it. That hasn't been a trivial process to go through the different stages of showing that we could measure luminescence at all using the technology. So we can show growth curves in planktonic culture and within macrophages. So the next steps for this project are to, as I said, to kind of do these comparisons between these different strains within the M. ulcerans complex to see if they interact with macrophages in a similar or different way, and then perhaps try and understand those similarities or differences. And we're also interested in the host side of the equation. So what is it about the host response that either allows that replication or controls that replication?